Good afternoon. Thanks everyone for attending the Nature Repair and Climate Adaptation stream. Our next speaker is Renee from the Big Scrub Rainforest Conservancy, and she'll be speaking on genetic rescue of the Big Scrub Rainforest. So welcome, Renee.
Just click through. Oh, is this working? It's not working. It's just me. So I go to the next one. Aye, that's good, we're good. Okay, hello and thanks for coming along to my presentation today. I'd first like to acknowledge the traditional owners of the lands on which we're meeting today, the Conservancy people. I'd also like to acknowledge the traditional owners of where I live and work, the Widjabul Waiabul, the Arakwal and the Nyimbul people of Bundjalung Nation. I pay my respect to elders past, present and emerging, and extend that respect to any First Nations people we have in the room today. I'm Renee. I'm an environmental scientist and General Manager for Big Scrub Rainforest Conservancy. You may also know us under our former name, which was Big Scrub Landcare. I'm excited to share my presentation today about the Science Saving Rainforest program. It's a large and ambitious multi-species landscape scale restoration project with aims to improve genetic diversity in the critically endangered lowland subtropical rainforest, in particular in the Big Scrub Rainforest in northeastern New South Wales. The Science Saving Rainforest program uses the latest genome science to inform us of the individual trees or populations from which to collect propagule material from that will improve the genetic diversity for 60 rainforest plant species. So these include 30 threatened species and also 30 key structural species that make up the canopy and other key structural components of the rainforest that give it its unique microclimate. It will facilitate those 60 species to adapt to emerging threats such as climate change, new pests and diseases. For anyone who's not familiar with the Big Scrub, it was once a 75,000 hectare continuous patch of lowland subtropical rainforest in northeastern New South Wales, stretching between today's towns of Ballina, Byron and Lismore. To paint the picture, think of a blanket of thick, lush rainforest as far as the eye could see, dotted with tall sentinel emergent figs above the canopy, towering red cedars, buttressed yellow carabines, bangalow palms, epiphytes, orchids, brilliantly coloured rainforest birds. You get the idea. The Big Scrub is an ancient Gondwana-descended rainforest with tree lineages that date back many millions of years. It's described as having more Gondwana rainforest history and diversity in the living flora than any other comparable region on Earth. It's home to 38 threatened plant and 61 threatened fauna species and has the richest biodiversity of any ecosystem in New South Wales and second only to the tropics. So yeah, it's a big deal. Tragically, the Big Scrub was systematically destroyed. Its destruction kicked off in the mid-1800s by the cedar getters, chasing the red gold, the magnificent red cedar. In the latter half of the 1800s, the scrub, which was a derogatory term, was required to be cleared when the land was divvied up and allocated to new settlers to the area. By the turn of the 20th century, only 1% of the former glory of this rainforest remained in approximately 80 small, isolated pockets spread across the heavily fragmented landscape. Sadly, it's assumed that a number of species were completely lost during this period of mass destruction. The remaining Big Scrub remnants are depicted by the bright green patches in the map on the right of the screen, with the bright pink outline, the original extent of the Big Scrub rainforest. For the 400 plus plant species that remain within the Big Scrub remnants, fragmentation has cut off much of the previous gene flow that existed between plant populations, and over time genetic diversity has declined. Over the past 30 years, Big Scrub Rainforest Conservancy has planted and facilitated the planting of more than 3 million trees to help restore the Big Scrub rainforest. Yet seed for much of the planting stock came from the same easy to collect trees year after year. and this has resulted in a narrow gene pool within those restoration plantings. So why does genetic diversity matter? When it comes to subtropical rainforest woody plants, the majority of species rely on cross-pollination as a reproductive strategy. So they need another plant of the same species to cross-pollinate with. Unfortunately, the more fragmented and isolated the population of a particular species, the more likely that that population will become inbred. Inbreeding causes some rather serious issues. So there's a reason why we as humans don't marry our cousins. It's the same deal for plants. So this is as sciencey as I'm going to get here, but a quick recap on high school genetics. So there's one allele, which is a form of a particular gene that's inherited from one parent, and another allele that's inherited from the other parent. There's dominant alleles and recessive alleles. So in the diagram on the left of screen, the dominant alleles are represented by the black capital A's, and recessive alleles are represented by the red lowercase a's. So inbreeding increases the likelihood of the recessive alleles coming together, so the two little red A's coming together. Inbreeding can then lead to a situation called inbreeding depression, and that is a decline in the biological fitness of a population as a result of an increased expression of harmful recessive alleles. So this can result in lethal and sublethal issues that may include impeded growth, fewer seeds being produced, plants more prone to disease, and lower survival rates. Reduced genetic diversity can result in reduced adaptive potential within individual species as well as within the overall ecological community. How will critically endangered ecological communities such as the lowland subtropical rainforest have a hope of persisting in the future if they can't adapt to new challenges such as those presented by climate change, new pests and diseases? Frankly, without a diverse gene pool, individual species and entire ecological communities may find it very difficult to adapt to future conditions. Places such as the incredibly beautiful big scrub rainforest will quietly slip away, and its gone one and heritage will be lost forever. So, go back one. Didn't work. That's gone. Anyway, genetic rescue to the rescue. There was meant to be something that came up there. So this is where our Science Saving Rainforest program comes in. In partnership with the Research Centre for Ecosystem Resilience, or RESA team, from the Botanic Gardens of Sydney, Dr Rob Koyman and Firewheel Rainforest Nursery, Big Scrub Rainforest Conservancy's Science Saving Rainforest program was launched in 2017 and is investigating the genetics for 60 lowland subtropical rainforest trees and woody plant species. To date, more than 10,000 leaf samples have been collected across the entire distribution range for 32 species by Dr Rob Koyman and Associates, with leaf sample collections also underway for the remaining 28 species in the project. This has included leaf samples being collected across each species' full distribution range, which in some cases is covered from southern New South Wales all the way through to far north Queensland. Leaf samples have their DNA extracted, sequenced and analysed by the geneticists at the Botanic Gardens of Sydney's RESA team. This allows them to identify the similarities and differences in the DNA of various populations across the full range of each species and to determine each relevant genetic neighbourhood. So surprisingly, genetic neighbourhoods can be much larger than we first anticipated. The RESA team can then provide us with guidance on the best 25 to 30 individual trees or populations from which to collect propagule material that collectively captures 90% of the genetic diversity for a particular species across the northern New South Wales genetic neighbourhood using common alleles. They also advise us where to collect a small amount of propagule material from within a future climate-matched region that has a climate that currently is similar to what we expect the big scrub to have in the year 2070. So this will help our big scrub populations adapt much easier to future predicted warmer climates conditions. So with our partners, Firewheel Rainforest Nursery, we then grow the cuttings on that have been collected from the 25 to 30 identified trees or populations from each species until they're big enough to plant out in our living seed bank plantation. Seed produced once the plantation trees have matured and cross-pollinate will then be used to provide genetically diverse planting stock for future restoration projects to increase genetic diversity across the big scrub remnants and existing restoration plantings. It'll also increase resilience and adaptive potential to future threats such as climate change, new pests and diseases. So here's a list of the 60 species that are being included in the Site Saving Rainforest program. The 30 key structural species are on the left and the 30 threatened species are on the right. Threatened species were fairly obvious to include in the program given their current risk of extinction. However, we also felt it important to include the key structural species that are listed here. Even though they're not currently threatened with extinction, they do make up the key structural components of the critically endangered lowland subtropical rainforest ecological community. It's important to give some foresight now into improving the genetic diversity in these key structural species. Otherwise, we risk losing the key structural elements that give the rainforest its unique microclimate and their important role in providing habitat for other species that already are threatened with extinction. The genetics work for these 60 species will inform the development of our living seed bank plantation, which I'll discuss more in a later slide. Okay, so before I continue, the next 3 slides were developed by Richard Diamond from the Botanic Gardens of Sydney. I'm not a geneticist, so I will try my best to explain these in layman's terms. One of the key reasons for collecting leaf samples across the entire distribution range for a species is to identify genetic clusters or genetic neighbourhoods. The funky looking diagram we have here on the right of the screen is called a genetic splits tree. Each coloured dot represents a leaf sample from an individual tree that's undergone DNA sequencing. The graph on the left is a result of principal components analysis statistics being run on the DNA. And it just helps us tease out how different and similar each leaf sample is compared to other leaf samples. The genetic splits tree here on the right groups the similar leaf samples' DNA results by the different colours. So the further away they are on the splits tree, the more different they are. To put this in better context here's some results for the Diospyros pentamera, the myrtle ebony. So you can see how the genetic splits tree correspond with the different areas on the east coast of Australia map. The groupings of different colours on the map indicate the different genetic neighbourhoods. So if you look at the genetic splits tree, those northern populations are very different to the southern populations given how far away they are. Now, if we hone in on just the southern populations, we start to get a better resolution of the different genetic neighbourhoods in this area. The big scrub rainforest sits within the dark blue genetic neighbourhood. So you can see here that the genetic neighbourhood spans across either side of the northern New South Wales and Queensland border. Of course, nature doesn't care about state borders, but it causes us some logistical challenges when we have to deal with the various state authorities. Okay, so also as part of the sampling guidelines that we get from the Botanic Gardens of Sydney, after the genome analysis has been completed, we receive guidance on where to collect propagule material, that is cuttings or in some cases seed or wildlings, to capture the collective 90% genetic diversity for that species in the northern New South Wales, southeast Queensland genetic neighbourhood. For the example given here on this slide for Argyrodendron trifoliolatum, which is the white bouillon, we were advised to collect propagule material from 5 individual trees in populations located in each of 5 subregions. So in this case, just ignore the yellow polygon along the coastal fringe. It's the southerly extent of a more northerly located future climate matched area, and it's not really relevant to what I'm explaining here, so just ignore the yellow. Each of the other colours, though, represent a sub-region. So here we're being told to collect propagule material from 5 trees spread across various populations in the blue polygon. Same again for the green polygon, the orange polygon, the red polygon, and the purple polygon. So 25 trees all up, spread across the various sub-regions. As you can see, we don't have populations over the Queensland border included in these guidelines. It doesn't mean that we haven't considered the Queensland population genetics for each species we have. However, propagule collections for Queensland populations will require different state approvals and are a work in progress for us. To ensure that we capture the exact same genetic makeup of the wild parent trees that we've been guided to collect from, this is particularly for the threatened species, best practice is to collect cuttings material from the parent plant rather than seed. However, wild trees are a bit like wild animals and they don't always do what we want them to do. So this means when we've got a species that doesn't want to grow from cuttings, we need to consider collecting seed or young wildlings. So we make sure that we stick to the recipe. We need to ensure that we collect propagule material from the correct mix of 25 to 30 individual wild trees as per the guidelines that we've been provided with. We also need to collect all the relevant metadata when collecting propagule material, including the precise location of the tree that we've collected from, site conditions, soil type, aspect, etc. This metadata stays with every propagule throughout its entire life. So from its time of collection from the parent tree, through its journey through the nursery and to its final planting position in the living seed bank plantation. So developing propagule collection protocols before any of the propagule collections commenced was actually really important to ensure that everyone involved knew exactly what they were required to do. Our propagule collection protocols include information for our propagule collections team and the nursery team, such as all the metadata that's required to be collected in the field, examples of what good propagule material looks like, as well as what unsuitable material looks like, recommendations for ensuring propagule material is kept in good condition on its journey through to the nursery, and field and nursery labelling requirements, etc. Our propagule collectors enter the metadata for each collection into an app that's been developed by the RESA team at the Botanic Gardens of Sydney while they're out in the field making the collection. The RESA app can actually be downloaded by anyone, and it's very useful for collecting a variety of field information about the site and the various species while you're out in the field. If you choose I'm collecting for myself, just on the bottom of the screen there, as you enter the app, it'll ask you for your email address. You then proceed through the screens capturing the relevant information that you want to capture along the way. One of the key things for our project was coming up with a field labelling system that was reasonably intuitive to understand and not too long, as it had to be printed on labels that are attached to each plant once the propagule material hits the nursery. So in the example on the screen here, highlighted in orange and the red circle around it, DC is short for Diploglottis campbellii, NN is short for the Northern New South Wales genetic neighbourhood. 3 is site number 3. We use site numbers for threatened species. However, if this was one of our key structural species, the more common species, then this number would indicate a sub-region. HB is short for the site name, which is in this case Hogan's Bluff, and C1 is short for Cuttings Batch 1. If we were collecting seed, then that would be S1 or S2 on the end, and if it was wildlings, it would be W1, W2, you get the idea. So this is just a short label, and the full metadata sits in our master database, and it's tied to that short label. I'll show you that in the next slide. It also, most importantly, allows you to capture the field location details of the tree you're collecting from. by recording the lat long and the altitude and accuracy of that reading in the app. At the end of the day, our collectors export the data to their own email address. The export data comes through as an Excel spreadsheet, and the collectors then have the opportunity to look through that spreadsheet and make any corrections before they send that on to me. All of the metadata is captured in RESA app, is saved into our master database, which I've just given a small snippet on the bottom of the screen. This master database seems tedious, and yes, it is, but it's essential to maintain and keep accurate so that we know where we're up to at any given point in time. We know the number of cuttings that we have, their pot size, we determine if recollections are required, etc. The master database is also connected to our nursery printer, allowing fast, accurate printing of labels for the 30 to 60 cuttings that are produced for each propagule collection. So it's a big job, but we've got a highly dedicated team, and it makes it quite a smooth and enjoyable process when everyone's on board. So a key factor in selecting a suitable site for our living seed bank plantation, where the genetically diverse mix for each of the 60 species will be planted, was that the site needed to be located on the beautiful rich red ferrosol soils of the Big Scrub footprint and be in an area that minimised bushfire and flood risk. So we've been incredibly blessed to have been gifted funds from a private donor to purchase a property at Maclean's Ridges for the living seed bank plantation. So it's smack bang in the middle of the Big Scrub footprint and just absolutely perfect for what we need. The plantation will be located on the gently sloping land on the far left of this top image. To prep the soil for the plantation, we sowed a cover crop last year which included a mix of chicory, plantain, daikon radish, vetch and arrowleaf clover. So this cover crop will help build soil structure and nutrition, enhance the soil's water holding capacity and improve microbial biomass to ensure we've got good nutrient transfer and cycling within the soil. So in the coming weeks, we're going to be planting the first trees in our living seed bank plantation, and we're super excited to get to this really monumental milestone. It's been years, yeah, it's cool. Thank you. So there's some guiding principles that need to be taken into consideration when designing a plantation layout for a project such as the Science Saving Rainforest program. So these include not planting one species in one big long row. Instead, they need to be planted in species blocks to maximise cross-pollination. The layout of trees within a species block needs to be randomised. The randomised layout should be developed before planting occurs so that it's a truly randomised layout within the block, rather than just planting trees as they become available from the nursery for convenience. So this ensures no bias in the cross-pollination between trees within each species block. In other words, we don't want 5 trees from sub-region 1 all planted together, then 5 trees from sub-region 2. In the schematic representation of the planting blocks on the left of this slide, the pink shading shows the randomised locations that need to be left open for the Queensland propagule material that'll be planted at a later stage. Again, we don't just tack that Queensland material on for convenience when it becomes available. It presents us a bit more of a challenge for maintaining these blocks, these species blocks, while these gaps remain open, but it's important to do to make sure that we get that overall randomisation goals for the cross-pollination. Another consideration is ensuring that species blocks, trees in the species blocks are planted within 50 metres, ideally, and no more than 100 metres from each other to maximise that cross-pollination. We are also undertaking research that will inform us of the best way to encourage pollinators to the site. So in general, many lowland subtropical rainforest tree species have small white flowers that are pollinated by invertebrates such as beetles, flies, native bees and wasps, and we're currently looking into the best host species for these that can be planted in the interrows and around the perimeter. There's been a number of challenges that we've experienced along the way since we started the project in 2017, especially as this project's the first of its kind on such a large and ambitious scale. Having a completely dedicated team has really made this much easier. As previously mentioned, rainforest trees aren't like camellias that have been produced by cuttings generation after generation. We ran a smaller pilot project with the Firewheel Rainforest Nursery early on in the piece to ascertain how many cuttings that were needed to ensure that we could allow for some failures but also met the minimum requirements for the plantation. So that pilot proved very informative and laid down the basis for the propagule collection work. But we've also found that over time, some trees within a species do actually strike better than others within that same species. So sometimes we have to collect seed or wildlings to hedge our bets when those are a little bit iffy. So any work that involves a translocation of a threatened species also requires a translocation plan and the issue of a scientific licence for that translocation work. So usually a translocation in New South Wales is done on a single species basis. We've got 30 threatened species, anyway, we've managed to do one translocation plan for all 30 species, and it's taken us a year to get that through approved. So take-home messages: start early on translocation plans. I'm almost at the end. So we've been fortunate to receive the backing of numerous New South Wales state government agencies, Saving Our Species and Environmental Trust. We're also fortunate to be receiving some additional financial benefits from DNA work and propagule collections that are being done for some of the threatened species from National Parks and Wildlife, as well as Department of Climate Change, Energy, Environment and Water. We also need to acknowledge the huge amount of in-kind work that's gone into this project from our project partners, the RESA team at the Botanic Gardens of Sydney, Dr Rob Koyman, Firewheel Rainforest Nursery. In addition, I'd just like to acknowledge Dr Tony Parks, our former president, who's now 95, for dreaming big and working tirelessly to put the plans in place for this project to get underway. We've received financial benefit from other and in-kind support from Big Scrub Foundation, Rekindle Foundation, Stone and Woods Ingrain Foundation, Brook Farm, Cape Burren Distillery, Flowhive, and a lot of other private donors along the way. Science Saving Rainforest program is a globally significant blueprint for the genetic rescue of not only lowlands and tropical rainforests, but also for other threatened and degraded ecological communities. I'm very happy to discuss the details of the project if you want to know more, so thanks so much for listening.
Thanks very much, Renee. We've just gone a couple of minutes over, so we won't have any questions. But if you're wanting to catch up with Renee, it's afternoon tea time now, so that's back. A bit of a walk back to the main room. Thank you.